All right, here we go. Welcome everyone to Significant Figures. Happy Science Week. My name is Tom, and I'm the host of STEMpunk podcast, and we are doing a live show across the internet where we're going to talk to three amazing people who you can see right now and who we'll introduce very soon. And they're going to be talking to a fantastic mystery guest who's off in the green room at the moment. And we're going to have a lot of fun along the way. This is how it's going to work. Firstly, just going to introduce everyone because they're awesome and you need to know who they are and why they're so cool. Then we're going to ask some questions in round one, which is called 22/7, which is 22 yes-no questions in 7 minutes. Yes, it's a maths joke. The next round will be called the jargon game. And we're going to be talking about some jargon from our mystery science field and trying to work out what that is. Our panelists will try and figure out what those jargon and acronym terms are. Then we'll have a game called Two Truths and a Lie, where our scientists will tell us two truths and one lie, like it says on the tin, from their field. And we're going to have to figure out which of those are the truth and the lie. Guaranteed there will be some questions after that. And then after that, we're going to have a chance for our three panelists and you to start asking questions. You can ask questions at any time. Just put them in the comments for YouTube or Facebook, and they will come up, and I can pop them up onto the screen so we can see what the question is and ask our scientist those questions. There are a few things that I want to tell you. First of all, there are giveaways. So at the end of this podcast, I will send you a feedback form, very, very simple feedback form. And in there, you can tell me if you want some giveaways. It's quite literally that. Just give me some feedback and write your name down if you want a free giveaway. And the first 10 people from each of these shows will get something sent to them. What will you get sent to you? Good question. Well, it will be a shirt like this from Science Week, a hat from Science Week, a couple of stickers, which are kind of cool, and a few other things. That's fun. That's lots of fun. Anyway, so that's some giveaways for you. Start asking some questions. Lastly, before we get into it, I would like to tell you a very quick story. It's a story about my 7-year-old daughter. The other day, she was playing with her ushies, those things from Woolworths, and she lines them up and she teaches them. That's her game. She teaches them lessons. She's very good at it. And so what she did the other day is before she taught them anything, she just said, all right, guys, before we start, I just need to acknowledge the traditional owners of the land where we were on. And I thought that was super cute and super important. She realizes that her ushies are doing learning in a place where learning has happened for tens of thousands of years, as we are now. Me, I'm on Wongal land. And right now, in this location, for tens of thousands of years, learning has been happening, just like you. Anyway, are there any questions from our panelists? No? How about my question to you is, Alice, can you please introduce yourself very quickly so we know who you are?
I can, Tom. Lovely to be here. Thanks for the invite. I'm joining from the lands of the Gadigal and Bidjigal people of the Eora Nation today. My name is Alice Motion. I'm an associate professor at the School of Chemistry at the University of Sydney, and I love science, in particular chemistry, but there's room for many types of science, which is great. And I research different ways to connect people with science, amongst other things. Thanks for having us, Tom.
Annie.
Hey, my name's Annie Hanma. I'm 47 days away from submitting my PhD thesis in history and philosophy of science. So I do science and technology studies, a bit of sociology of science, and I teach science ethics and also space law. I spend a lot of time following around space scientists and basically trying to find out what they do and how they do it. So that's me. I also run the Space Junk podcast, if you've heard of that one, and Space Cafe Australia. Thanks, Tom. So excited to be here.
Yay. Okay, Joel, tell us who you are.
Thanks, Tom. Hi, everyone. My name's Joel Werner. I'm a science journalist at the ABC. I'm here on Gadigal land today. And yeah, I make science radio and podcasts with the great team at ABC Science. We have a new one out as of yesterday called Cosmic Vertigo, featuring Deadly Science's Corey Tutt and the wonderful Carly Noon. So once you subscribed to STEMpunk, go and subscribe to Cosmic Vertigo. But looking forward to hanging out with everyone today.
All right, thank you so much. Now it's time to start asking some questions for our mystery scientist. You can't see them, but I can. So we're going to start with some questions. This is round 1, 22/7. So we will get each of you to ask a question, and I'll start counting them. And I'll time for 7 minutes, and I'll give you some countdowns as well. And I want you to get as close to this person's field, area of research, as specific as you can in 7 minutes. Your time starts now. Alice, go for it.
Do you study things that are alive?
That is a yes.
Yes. Okay.
Ooh, all right. I think I'm up next. Do you study things from a lab?
We're going to go no with that one. That was this for a minute, but then it was a no.
Okay, but there's some uncertainty there. That's good.
I think there was a little bit of uncertainty. Yeah, sure.
Okay.
Joel.
Okay, I'm going to need you to be honest now. Like when you're at a party, remember parties for those of us in lockdown. When you're at a party, are people genuinely interested when you tell them what you do? Or are they just faking it?
Oh, emphatic thumbs up. Yes.
Oh,
Excellent. Okay, great.
oh, that's
Alice.
good to know. I would like to know the things that you study. Would you be able to carry one of them in your arms to a party?
Nice question. Yes, you can. That's the science version of does it fit in a bread bin? Go, Annie.
I'm so excited. Okay. I'm narrowing it down in my head. Do you use any special equipment as part of your research?
Thumbs up. Yes, thumbs up. 5 questions down, and you are 5.5 minutes left to go.
Okay. Do you ever have to do field work? Is there does your research take you out of the lab and into the world?
Yes, field work into the world. Yes.
Is there a potential for the thing that you study to seriously harm humans?
No serious harm to humans.
Okay.
Ooh, okay.
Good
All right.
question.
This is some seriously good questions. Okay. When you're doing your research, is it ever smelly or dirty?
Nice. No smell. Very clean, apparently.
Okay. Maybe a bit left field. When you submit a research paper, is it one of those papers that has 1,000 names on it? Let's say more than 20 names on it, or is it a smaller group of people?
That was a cheeky way of doing a yes or no question, but the answer is no to the question you asked. Okay, here you go.
I'm not sure what that means. What is it that? So was the question: are there more than 20 people on your research papers typically?
Yeah, it's are you taking a photo of a black hole and have hundreds and hundreds of authors, or is it kind of more niche than that? That's kind of what I was getting at. So it's a little bit more niche than the Event Horizon telescope's work.
Yes, that's right. Yeah.
Cool.
We've got
I would love to see someone turn up to a party with a black hole, in their arms. People would be genuinely interested for the moments before they fell in.
Alice, your go.
Can the things that you study move on their own from one place to another?
No.
Okay.
The answer is no to that. That's 10 questions. You have 3 minutes and 8 seconds left.
I think we're doing really well. Okay, so I'm thinking if they can't move, but you could carry it to a party and it can't harm a human, I'm thinking some sort of plant at this point. I don't know. Is that what we're all feeling?
No, that's a no.
Oh,
No.
not a plant.
Not a
Not a
plant.
plant.
Oh my goodness.
Have we asked if the thing is alive?
Yeah.
Has that? Oh, we have.
Yes,
That's
and it
definitely
was, right?
Okay. Does the thing that we're carrying to a party that definitely won't kill us, that is alive, is it furry?
Not furry.
Okay.
Does the thing that's alive that we can carry to a party that isn't furry live with inside another organism?
Yes.
Okay.
All right. I think
Ooh.
we're getting good
Okay.
now.
If
2 minutes left.
If I took this thing to a party with the group of people here and I sneezed on them, could I give this thing to another person?
Yes. Yes, you can. What a gift that would be.
What a gift.
So is this thing large enough that I can see it with the naked eye, or do I need equipment to see it?
Yes, you can see it with the naked eye. Yes, you can. You have 15 questions in, 1.5 minutes to go.
Oh, goodness. This is really getting trickier. So it doesn't cause harm to people, but you can pass it to other people. You can see it with the naked eye. It lives in different organisms. Does it have a long lifespan?
No, not a long lifespan.
No. Okay.
Oh my goodness, this is
Annie,
difficult.
off to you. 45 seconds to go.
Oh, okay. This is I'm
You
really
can
on the
pass if you
spot
like.
here. I'm gonna pass. Joel,
Okay,
you got something?
Joel, go.
I was just thinking, like, does this thing have feelings? That might be a bigger question than we have time for in this whole event. But let's
The
just
answer is no.
in the dark.
It has no feelings. Alice, 20 seconds.
Can you kill this thing with a medicine that has already been developed?
Yes. Yes, you can.
Why would you
10
want to kill
seconds.
something that cause harm?
If it doesn't feel anything, it's like, well, just for fun, maybe.
Does anyone have a final question for the last 2 seconds?
Does the thing you study help to turn food stuff from one type to another?
That is a no, and that is our time. We got through around 19 questions. I think that was 18 or 19. We ran out of time. So now I'm going to ask you, what do you think? What field? What area of study? Have a guess. And then
Look,
we will
it's not astrophysics. I think we ruled out black holes pretty early on. Alice, I feel like you seem to have the best idea of what was going on.
I don't know. Like, I was thinking that the person might study something like a bacteria or a protozoa, but one that isn't harmful to humans. That was the way I was thinking about things. I thought they could be a biologist or a virologist or something like that.
Yeah, I was thinking like gut biome kind of stuff. Yeah.
Yeah. I suddenly thought it was something like lactobacillus, like something that was, from that we could be looking at, that's where my food question came from. They were looking at a bacteria that could, help to make sour foods or fermented foods, if that makes sense.
That's pretty
we have
cool.
someone
My
who
brain
says
was going other ways. I was like, maybe maybe what we're catching here is feelings, and maybe they do something to do with emotions.
Aha.
Well, I think it's time to meet our mystery scientist, and we are going to meet them right now. It is Michelle Demers. Woo! There you go.
Aha.
Hey!
Hi, Michelle.
Hi, Michelle.
It is a huge deal to try and hide you from people. But welcome. How did we go? Tell us a brief thing about what you do, Michelle, and it's absolutely wonderful to see you.
Ha! Classic mistake. Muted my mic and didn't unmute myself. Welcome to Zoom World, people. Yeah, thanks for having me on the show, and I'm happy to be here. Yeah, interesting questions. Sometimes you guys were close, but not quite. So what I study is not a plant, but it's a fungus, which is something that a lot of people tend to kind of miss. So in detail, what I study is a pathogen, but it infects plants. So when you said, do I study plants? I thought, maybe, but like not quite, because I really focus on the fungus. So I study the rust fungus, which you can see from the naked eye, because the symptoms is it gives plants these rusty orange colored spots, which is why we call it rust, because it looks like your plants have kind of gotten rusty. And so what this is a plant pathogen. So it infects our crops like, wheat, barley, rye, and oats, and can cause, food security issues, famine in the worst case. And so in a bit more detail, I actually do need specialist equipment. I use supercomputers. I'm not sure if that's what you had in mind for specialist equipment. But I use supercomputers, and I actually study this fungus in a field called bioinformatics. And so what this is a blend of computer science, biology, and statistics. And I actually do genomics. So I study the DNA of this fungus, and I use a computer to basically upload the genome of this fungus and then use, computer techniques and machine learning to sort of understand the genome and how it works and how this thing is actually infecting our plants.
So we were very close then, like super close.
Yeah, like almost there.
Yeah, fungus is kind of like a feeling.
Kind of. Well, I mean, like maybe
no
they have
holes.
feelings. Yeah, you got that. You got that right. As for the, are people interested when you go to a party? Depends how much you can sell it. Because if you say, what would you do if there was no beer? Because you need barley to make beer, and if you have rust attacking your barley, you have no barley and therefore no beer. Yeah, people kind of tend to care. But if I say, oh, I'm a computer nerd who like hacks fungi, everyone's like, So it depends on how you sell it.
Okay, if there are no questions for Michelle before we move on to round two, I'll say that another way. Are there any questions for Michelle before we go into round two? No,
Michelle,
we're about
how
to
you
learn.
doing?
One.
Oh, no. We all had questions. Alice, you go first.
I would love to know, why is it called rust, Michelle? Is it because of how it looks, or is there some reason why we call this plant rust?
I know it's just the color. So it has I think it's called cytochromes, which are pigments that are in the fungus, and these pigments actually serve to protect it from the sun. That's actually really similar to a lot of pigments you find in like fruit, like blueberries and things that give things color. They're usually protecting against the sun, and the ones in the fungus happen to present as orange. And so, yeah, when you look at plants, the most common description that people give to these plants is they say they look rusty, because, yeah, you've got this kind of deep orange color kind of in splotches all over your plant, and it kind of looks like, what might have happened to the bottom of your car if you've ignored it for a year. But in this case, it's happening to plants. So, yeah, that's why it has the name, because it has that rust-colored orange, and that's how you can tell that it's this particular fungus.
Thanks, Michelle.
Annie.
Oh, I was just going to ask how you're doing, Michelle.
Oh, yeah, I'm doing all right. So I hear you're submitting your thesis, Annie, in 47 days, you said?
That's right. That's the plan.
That's exciting. I submitted mine about 47 days ago. So, hey, see you on the other side soon.
For the last stretch, any tips?
Hang in there, coffee. The light at the end of the tunnel is there.
Awesome. All right, Tom, should we get into the next round?
Yeah, let's do it. Now, I just wanted to mention a few people who have made some comments, which is really great, and I appreciate it, and keep it up. So Stuart Mai has said microbes before. Didn't get a chance to display that. And Sydney Critic was there saying, I was thinking fungus or algae. So that's pretty cool. That's very good that we're on track. All right. So let's go into our next round, which is called Jargon Game. Here we go. So what I'm going to do is I'm going to read some jargon terms from Michelle's field, and I want you all to guess what they are. You can confer and you can chat with each other and have a bit of a chat. We'll do this about 10 times, so we'll move pretty quickly. But here we go. The first one is variety. In Michelle's field, what does that mean?
It's the name of the party you go to when the variety party. No, that's not right.
Keep going. I'm interested. I want to see where this is going.
Everyone wears a costume and they must bring a balloon that has to be suspended from their ear at all times. No?
So this term actually has a synonym. So you can say variety, but you can also say cultivar, and it means the same thing. Does that spark any ideas?
Is it only cultivar if you're in a certain region of France?
If you're in the cheese industry, yes.
Okay.
But here, no.
Is it something that relates to the type of species that you're looking at? So if you're thinking about a way of describing the species you're looking at, Michelle?
Yes, you're close. You're very, very close. Now
tell us what variety is.
Yes. So so this actually refers to the plants, which is the host of this pathogen, right? So it's a fungus and it infects plants. And the thing that it's infecting is called a host. So variety it's sort of a taxonomic classification. So, order, family, genus, species, that thing. It's actually one step below the level of species, which is typically seen as the lowest level, right? Homo sapiens, Homo, humans, sapiens, species. It's one level below that. And so people use this in the plant world because often plants will have very specific characteristics, and these characteristics are measurable and are standardized throughout different varieties. So for example, you might have a wheat variety that's really good for bread making. So the seeds of that wheat will be very high in gluten. And so if you're, selling off your wheat to a bread maker, you want to make sure that it is a variety containing high gluten, for example. But these varieties are also important in my field because a lot of them too have very specific disease resistance genes. So you want to make sure that when you're planting a field of wheat and you know that there is a lot of this rust fungus in the area, you need to make sure that you are planting a variety that has resistance to this fungus, because not all varieties will.
Okay, excellent. Next term, which will be dikaryon.
Is that it's what you ask yourself when you've got a really difficult question and you're wondering whether you should continue?
Do I care? Do I carry on?
Do I carry on?
Nice.
Hey, can I ask you to spell that?
Yeah.
Yes.
Go for it, Michelle.
Sure. It's Dikaryon.
Okay, there's a K in there. I'm thinking this was from an ancient Greek root. Alice, I'm looking to you for this one.
We were joking about this before. I think it's something to do with something that's in the genetic material or the nucleus of a cell, but I don't know too much more than that.
You're on the right track.
Di, I'm thinking, might mean like two.
Mm-hmm.
Yep, it's good. Very good.
So there's two things happening somewhere in the nucleus of a cell, potentially.
Oh, you're so close. You're so close.
Two things happening cellularly. Joel, you've been sitting this one out. Have you got any thoughts?
I've almost turned it into an anagram for some, like, you are kind almost. I've just been making an anagram with the word cell. No use at all.
I think we've just...
Do you have
dikaryon? Yeah.
Oh, that's a good one. Is it when do you have like two nucleus from different organisms fusing, or something happening about two of them coming together or splitting apart? So I don't know.
You're
like
so
a hybrid?
You're so close.
Tell us what dikaryon is.
Okay. Take the leash off. So you guys are so close. So a dikaryon is a type of organism where you have two separate nuclei inhabiting a single cell.
Okay.
That's a dikaryon.
Okay.
So this is actually a really common thing in fungi. So when I first, started studying these things, and everyone's like, hey, if you're doing genetics, you should probably be aware that instead of having one nucleus with all of your DNA, like, most animals, because we tend to view animals as being the standard, which, in nature, animals are definitely not, what's normal. We don't know what normal is because we don't know half of what's out there. But anyways, we tend to view having cells with one nucleus as normal. But when we move into fungi, what's actually quite normal, especially for a class called Basidiomycetes, which is most of like the large mushrooms that you see are usually sort of within that order. A very common thing is, they have one cell with 2 nuclei, and they have half of the amount of DNA in each nucleus.
Cool.
Yeah, and so it makes it a very interesting problem when we work with this computationally as well, because on a computer, we tend to represent a genome, as pretty much all of the DNA in one nucleus, because that's how, again, with animals, which is what we view as normal, that's how we tend to do it. And so one of the great challenges that actually we have working with genomes of these fungi is we actually try to separate those 2 genomes by their nucleus. So we actually try to assemble the DNA into 2 separate genomes to represent these fungi accurately.
Wow. So you have to do twice the amount of work when you're studying fungi using supercomputers.
Yeah, it's this whole process. So there's a funny process. I don't think this is in the jargon list, but it's called haplophasing. And this is the process whereby you take that genome and you try to split it into those 2 separate nuclei. And if there's any real computer nerd people out there, like, that is an NP hard problem. Like, it is a very computationally difficult thing to figure out. And actually, it's getting easier because we're actually getting better technology. So one of the newer genome sequencing technologies we have actually takes into account the spatial information of the DNA. So it's not just what the sequence of the DNA is, it's also where in the cell is it located, which pieces of DNA is it located next to, right? Because all the DNA in one nucleus is going to be closer to each other than all of the DNA in the other nucleus. And so you can actually use that spatial information to say, hey, these bits are found together, so this is probably one nucleus. These other bits are all found together, so this is probably your second nucleus. And with that, it's making things a lot easier.
Is there like a dominant nucleus in this situation, or are they equal, all things the same?
Pretty sure that they're equal. Pretty sure. Can't say 100% because there's still a lot we don't know, but a very good question.
Saturday afternoon is like, pretty sure enough for Saturday afternoon.
On point. Yeah.
Okay, let's go to our next one. I'm going to skip a few. We're going to go to this one here, SNP. This is an acronym, SNP.
Pronounced SNP.
Yes, there you go. SNP.
Is it like still not processing when you're like hitting the side of your supercomputer, when it's like you've got the blue screen of death and little rainbows spinning? You're like, SNP.
I know there's usually a 4-letter word we use for that situation, but I won't say that here. It is a computational DNA term, though.
So it doesn't stand for something that's not physics.
So there's like a snip tool in audio editing where, like, you kind of like you snip out whenever I say in this, Tom will probably diligently go through and like snip out all of my ums. Is it a similar thing but for DNA? Are you snipping out bits of the genome?
Not quite. It's more of a 1-for-1 swap.
Okay.
Does the P stand for process?
No, stands for polymorphism.
Okay, all right, cool.
Because I'm sure that'll help you a lot.
Oh, Paul knows this one.
Yeah.
Apparently, Paul knows this. So, Paul, when you hear this, tell us what you think. In the meantime, we're just going to have some more wild guesses from these guys.
Got another good acronym for me?
Polymorphism. Does that help?
Is it single nuclear polymorphism?
Yeah, single nucleotide polymorphism, but that's, well done. Woo!
Wow, well done, Al.
Good one. Yeah, so
or should we say Paul?
Paul, yeah.
That's you, isn't it?
Thanks for texting me, Paul.
Nice. Yeah, so single nucleotide polymorphism, which is a mouthful, and there's a lot of them. So we just call them SNPs. So what this is this is when you have a DNA base pair change of one base pair, right? So DNA, you have, A, T, C, and G multiplied by a lot. So, for my organism, for example, I'm looking at 90 million pairs of these things, right? And you know that, between you and me, we don't look the same, right? We have differences in our genetic code. A lot of these differences come down to SNPs. So what a SNP is that in the same location in the genome, different individuals, say, will have a different letter, so where I have, ATT, Annie, you might have GTT, right? It's a single nucleotide change, polymorphism. Yeah, and so these are some of the changes in DNA that account for a lot of the differences you see between different people. This is why, we look different, for example. In the case of these fungi, these single nucleotide changes might be responsible for, say, making a gene that causes virulence to function properly versus making one not function properly. Because a change in the DNA will later on create a change in the protein that it codes for, and then changes in the protein might cause them to behave differently. So these changes are really, really important to track between fungi when we want to see, which ones are likely to infect who, and which genes are working and which genes are not.
Is that the reason, Michelle, that you've already submitted your thesis and I still haven't? Can we put it down to genetics?
Oh yeah, but see, here's the thing: is when it comes down to genetic things, there's always genetic bioenvironmental factors, right? And that can be really, really hard to pick out. So, maybe my genes made me finish earlier, but like, what about the environmental factors? When did you start, for example? Maybe you started later than I did. How difficult is your project? How easy were your test subjects to work with you, for example? Could be.
Fair enough.
Could be.
Yeah.
Never forget the environmental factors.
Is like G and T, is that SNP an environmental factor?
That's what Tom's sending round for us afterwards, I think.
Yeah, that's right.
Yeah, I could do with one of those. Yes, please.
Let's move on to our next jargon and or acronym term. This is INDEL, INDEL, INDEL.
So IN is the first acronym, and DEL is an acronym for the second word, which we squish into a single word, INDEL.
Okay.
Is there a deletion? Is the DEL for deletion?
Yep.
And does the N
correct.
stand for nuclear again? Or
Nope.
nuclear something?
Oh, and INDEL.
Oh, indel.
Yep.
See, I know what an outdel is. I'm all over that, but indel, I'm just saying, I'm not quite there.
If anyone in the chat wants to help me out,
yeah, I know.
Yeah.
We could do with another text, thanks. So it's not nucleotide, it's not nuclear, it's.
So think along the same train as DEL. DEL for deletion.
Oh, insertion.
Yeah, there we go.
Insertion, deletion.
Insertion, deletion.
Oh my god, I got one.
Yeah, you got one.
Yay.
Yeah. Yeah, well, I guess, Alice, you kind of guessed it pretty quick from the beginning, or at least half of it. Yeah, so indels, insertion deletions. So this is pretty much the next logical progression of the SNP, right? So a SNP was a single nucleotide polymorphism. And insertion deletion is the same thing, except with more than one base pair. So let's say I have ATT, and, Alice, you have G, you would have GTT, that would be a SNP. But a deletion, for example, would be I have ATT, you have nothing.
Mm-hmm.
Or an insertion would be, I have nothing and you have GGGC, repeat hundreds of times. That really does happen. Yeah, so this tends to cause much bigger changes than the SNPs do, because instead of just changing a one-for-one, you're either putting in a large chunk or you're removing a large chunk. And we can be talking of as little as 2 at a time. Adding into, removing to, but it can be hundreds, it can be thousands. In these organisms, you have, I'm trying to think of what it's called. It's escaping me at the moment. But you have regions that are repeats, right? So you have the same sequence of DNA, say, CTTAAG. I think that's the telomere sequence, right? You have that repeat. Some people will have that repeat 20 times. Some people will have that repeat 20,000 times, and it varies between each individual as well. And so this is just another mechanism that pretty much all organisms have in their DNA that can create such variation between individuals. And really, it's widely variable. How many base pairs you might have in one location and how many you won't because of these insertions and deletions.
Okay, let's do two more. We'll do two more, and then we'll go on to our next round. The first of the last two is effector. Effector. Effector.
Well, like a vendor or a conductor is the person doing the thing, doing the vend or doing the conduct. So is it the thing that does the effect?
Yes.
There's something making
You're on the right.
On something.
Yes.
Okay, with that stroke of genius, I'm going to hand over to the other.
That's how you do it.
Quit while you're ahead.
That's right. Yeah.
Yeah, I think it's the. I think it's that. It's the effector.
Could you put it in a sentence for us?
Oh, that's a good one, Annie.
Oh, okay. Fungi have a lot of effectors which help them to cause infection on plants.
Oh,
actually, bacteria have effectors that can cause infection in humans.
So an infection is when the effect does the in, whereas an effection would be the thing that takes the effect out. And the Latin root would be, I'm not going to say it because it sounds rude when you say it properly, but it means to do a thing. So I think it's a thing that does a thing outwards to infect. It effects.
Can I quote you in the introduction section on my next published paper, please?
I think the only way to make that better is. Sorry, go ahead.
There was a part of that sounded a bit like the Okie Kokie, the kids song. Like you put one thing in, one thing out. Put
the infect in, you put the infect out.
The only thing that made that answer better is if you ended with man. You put the thing in and then out, man.
Right? So I don't know. How close am I?
Is it a word or an abbreviation, Michelle? Is it
it's a word.
It's a word. Okay.
It's a word. It's a jargon word.
Yeah, like is it to use a metaphor, is it kind of like if the fungus has an entourage and like each of the entourage has their own kind of specific role and like their own like kind of role within the group, and then they're just kind of going to the club, which is the side of the infection, and they all like you talk to the guy in the door, I'm gonna go and get some drinks, and then that's how the infection happens.
Oh my god, you're actually really close with that.
You're a genius.
Wow, that is great. I'm gonna use that next time I have to do an event with effectors. Yeah, right. Yeah. So what effectors are is they are proteins. Well, actually, they don't necessarily have to be proteins. I usually think of them as proteins. They're any kind of molecule that the fungus has that promotes infection. So they're like the fungus's like wingman that distracts the guy at the door to the club and lets the fungus sneak in without paying entry. Yeah, so I'll get a bit more technical here. So the plant immune system, it's not adaptive like ours, right? So when we get sick, let's say like one of us catches COVID, because that's the thing everyone's talking about these days, right? Provided you have a healthy immune system, you're gonna get sick for a while. And then your immune system is going to kind of figure it out. And it's going to be like, oh, okay, like that's what this thing is. Here we go. And then it's going to create new antibodies. And then it's going to, kick out the virus and hopefully you'll be okay, right? Best case scenario. Plants can't do that. So our immune system, is adaptive. It learns, but the plant immune system doesn't. So I think of the plant immune system like a spy movie. Like just let's think of like Ocean's, is it Ocean's 12 or something? Where they've got like that golden egg, and then you've got all these tripwires that are in the museum and everything, and that's what it does, is it sort of monitors like the plant space, and it just kind of sits there and waits and watches, and the only thing it can do is recognize something that is not self. Right, so what it will do is it scans the plant space for molecules that don't belong in the plant. One of the big triggers, for example, is something called chitin. Chitin is like it's a sugar protein, it's a sugar, sorry, that's found on the outside of fungal cell walls, and plants don't have it. So if the plant picks up chitin and it's like, hang on a second, like you shouldn't be here, and then it just throws out the alarm, all the bells and whistles go off, and then the immune system goes into full drive. So what the fungus is a hacker, right? Basically what it does is it goes in and it has to deactivate the plant immune system so that it can then sneak into the plant and steal that golden egg, and the plant has no idea that anything's even inside of it. And so that's what these effectors do, is they go in and they either block the plant immune proteins, or they trigger them, or there's just so many different mechanisms that they have to basically take that immune system, hack it, and shut it down. And so then when you have essentially a very infected plant that looks rusty, in this case, the plant immune system is just completely compromised, and it doesn't even realize that it has a problem. And that's what these effectors do. And that's sort of my job, like as a bioinformatician. And this is the whole reason why I'm hacking the genome of the fungus, because I'm trying to find these effectors, and I want to find what they do and how they work, so that we can come up with better technology to be able to stop them and keep our plants healthy. So this fungus is basically a hacker. The effectors are kind of the tools that it uses to, shut down the alarms and the tripwires. And I, as a scientist, I'm trying to hack that hacker to try and figure out how does it work and what can we do to stop it.
I think not only was Joel tantalizingly close there, but also we're getting this deep insight into how lockdown is affecting Joel. So far, he's mentioned a party, going to a club, and GNT. And Joel, we're feeling you, I think.
I am jonesing to leave this apartment. Like, I am just like, in my imagination is just full of the outside world. I like the idea that sort of by oceans, whatever, 18 or something, it might actually be fungus infects plants. Like, that might be where they're at. I'd watch that.
It's molecular warfare. It's exactly what it is. It's genetic warfare that we're dealing with here. It's, you got to have the right genes in the plants that can actually recognize the effector genes in the fungus, and then the fungus evolves its effector genes to one up on the plants and be able to hack it and shut it down. And then the plant evolves new resistance genes to one up the fungus and find the new effectors and trap them and kill the fungus. And it's just, one is just constantly trying to out-evolve the other of these effectors from the fungus versus the resistance genes from the plants. It's an arms race. That's been going on for thousands of years.
So could a GNT kind of be like an effector that allows Joel to put on a strobe light and imagine that he is at a club?
Why not?
By dulling the senses a little bit. And
I will run an experiment later on and let you know, Annie, how it goes.
Great.
Please tell us in detail.
Let's do one more jargon and acronym term. We'll make this a quick one, and then we'll move to our next round. And this one is hypersensitive response. I think this is going to be fairly familiar to a lot of people. What does that mean in Michelle's field?
Is that when I watch the press conference and start crying?
No, that's not hypersensitive. Right, it's just appropriate.
Oh, yeah.
Is it like when someone gets called out on Twitter and like goes on a massive, like, has a big cry about it?
So I'll give you guys a bit of a clue in this sense. Think of it like kamikaze.
Oh,
is it like when when like the immune system gets hit by something and it overreacts and then starts attacking itself?
That's like the human version of it, kind of.
But in a plant, just to be clear.
But in a plant, and it's a good thing in a plant, whereas in the humans, it's kind of a bad thing, I think.
Okay. Alice,
so tell us.
I was going to guess that it's something where, so if it's a good thing, it means that with a very small indicator or amount of something that's bad for a plant, it sort of sets up itself to defend itself against whatever's coming by going into overdrive.
Yep, nailed it.
There you go.
Nailed it. Yep. So the hypersensitive response is what we call, it's pretty much another name for like the standard plant immune response, to be honest. So, right, you've got these plant resistance genes and they're monitoring that space for something that shouldn't be there, right? as soon as it finds it and recognizes it, it stimulates what we call the hypersensitive response. And so how this works in plants is you take yourself down and you take your enemy down with you. So what will happen is you have, say, a fungal spore that's snuck into a leaf, right? And this is for plant best case scenario. The cells around that site of infection have gone, oh, hang on, something's not right. You shouldn't be here. Those cells immediately die, right? And the goal of the plant is to be hypersensitive. You want to kill yourself as quickly as possible. But here's the catch, only in that localized region. Right, so this fungus needs the plant cells to be alive in order to pretty much mooch off of it, right? The fungus tries to just steal all the plant's energy and use it for its own nefarious ends. And so what the plant tries to do is kill itself off, but only in the cells immediately surrounding that fungus, so that if the plant cells are dead, the fungus dies too, but the rest of the leaf is fine and completely protected. And so often, you may have actually seen this if you've ever, gone for a walk outside. We're still allowed to do that, actually. That's right. If you go for a walk outside, and if you've ever looked at the plants, and sometimes if you look at leaves, you'll see like these little patches of like, dead tissue. That could very well be actually a successful hypersensitive response where some nasty pathogen landed on the plant and the plant went, oh, hang on, this isn't good. You shouldn't be here. Kicked in that hypersensitive response, killed only those cells around the site of infection, and the rest of the leaf is protected.
Very cool. Thank you. Okay, so now we're going to move on to, I always hate the transition between, one question and the next. It sounds very cool. That sounds insincere, but it's not. Very, very sincere. Anyway, let's
Yeah, very cool.
It needs like some music, like a fog horn.
That's editing afterwards. I'll do that. So here's our next round. It is called Two Truths and a Lie. What I'll do is I will read out 2 truths, 1 lie from Michelle that she sent me, and it's up to you to work them out. You can confirm. All right, so here they are.
I'm gonna poker face this one.
I'll use the exact sentence you tell me. So it's not me when I say I, it's Michelle. Okay. I got food poisoning on a train going through Kazakhstan. I spent my time being sick in the bathroom where the toilet opens directly onto the tracks below. That's number 1. Number 2, while doing remote field work, I drew the shortest straw and had to pull the leech out of someone's eyeball. And 3, I got chased by an aggressive cassowary while doing field work in northern Queensland. Our research team scrambled into trees for safety, and we were stuck there for 4 hours until they left. There you go. Which one of those is not true?
So we have spewing on a train in Kazakhstan onto the tracks, a leech in the eyeball, or a cassowary in 4 hours in a tree.
Yep.
Sounds like an EP.
Yeah, this is Triple J Unearthed right here. The Kazakhstan train that opens onto the tracks, that sounds legit. Just from a logistics train perspective, I have been on trains that have that does check out, but just purely from a train perspective. Whereas I have some questions about the leech in the eyeball, because don't leeches go for blood? And there's not a whole lot going on in an eyeball, from what I understand, having done a dissection once in high school. What do you reckon? What do you reckon, Alice?
Yeah, I think, it was a special kind of leech, but I would like to hear a little bit more about that. And then I also have some questions, Annie. So, Michelle, are we allowed to ask questions, Tom? Is this permitted?
Yeah. Ask some questions, but Michelle will probably be vague in her answers.
What do we think about the eyeball? Should we go with that one first?
I think that had the least detail, just in terms, I think the Kazakhstan train, if you're making that up, you don't need to add in that you're spewing on the tracks. Do you know what I mean? If that's the lie, then why go to that detail? And the cassowary is kind of the same. 4 hours is that's a very specific thing to lie about. Whereas the leech and the eyeball just seems a little undercooked for me. It's maybe could have done a bit more work making that lie up. So I'm happy to go lie just on that basis. But...
So Joel, I'm so with you on this one. The cassowary had so much detail that I thought this has to be true. And if it's not, then I'm going to question everything that I think I know about Michelle and about people and about life and about relationships.
Oh my God, Alex, you're okay.
But yeah, I feel like the
Existential
Cassowary
crisis.
one is definitely true. But if we're allowed to ask some questions, I would like to know, on this train trip in Kazakhstan, where were you going from and to?
Good question. Where was I going? Give me a second.
Convenient not remembering of facts.
Astrakhan to Aralsk.
Oh, okay. Yeah.
Is there
And
a
what were you
train line
eating?
there?
I would also have accepted that it was the party train that just went round in the circle, and hence why you got so sick. And sorry, I'm bringing it back to parties again. Like,
Yeah.
this is clearly my subconscious at play.
Yeah.
Stuart
It's that
says
one-
that that's the east-west. It goes east-west.
Okay. I mean, you know, that checks out with my knowledge of the geography. But I think, you know, Michelle could answer that one. So that one's, you know, I think I'm with Joel, and I think Annie was going this way too. I feel like it's the leech that's the red herring or the red leech.
All right, let's
Let's
lock
lock it
that
in.
in. Yep. And Michelle, would you like to tell us which one of those was not true?
You're all wrong.
No!
Ah,
The fake one was the Cassowary.
Ugh.
Good
There was
lie.
so much
Alice-
detail.
So
you
that's
just
what I-
fractured
Alice's
everything now.
soul.
I know.
Existential crisis not averted. No, that's kind of what I wish happened, because at least it would have made a cool story. The truth with the Cassowary is that it walked into our campsite, or sorry, it wasn't a campsite, our research site. It walked into our research site. Everyone panicked, screamed, ran for the car, started shielding ourselves behind the doors and ready to jump in. And then it just like calmly meandered through our campsite and just walked away, and we never saw it again. And it did not care.
That's very tricky though, because there
Was
was a
not
Cassowary.
aggressive. There was a Cassowary. It was not aggressive. It just walked through, and we never saw it again.
Hmm.
I don't even know if I can
And-
believe that now.
So yes,
I kind of have to know about the leech in the eyeball.
the leech in the eyeball. Yes. So this was on that same trip. So this was actually so when I first came to Australia, I came as an exchange student, and then I fell in love with the place, and then I returned as a dirty backpacker for a year. And when I was doing the backpacker thing, I ended up doing a lot of I ended up actually doing a lot of science and research jobs, which is actually really cool. I actually never did the fruit picking. It was always like, again, going up to northern Queensland and helping someone with their field work, or, working with robots to do really high throughput DNA extractions. I had some really cool backpacker jobs, actually. And so this was a fieldwork trip, and we were sampling tree species from the Atherton tableland. So this is really high elevation rainforest. My God, the terrestrial leeches, they go for literally like anything they can find. I wore a big pair of gumboots like up to my knees the whole time, whether it was raining or not. And I'm probably the only one who avoided having a leech bite on my ankles. Not to say I didn't get them on my face, but that's another story. So when you're in newer forests that have been more recently a clear-cut newer growth forest, you do get a lot of terrestrial leeches. But when you are in an older growth forest, so one that has been undisturbed for a longer period of time, the leeches are intense. They were everywhere, and they're very, very light, right? So the terrestrial leeches, like they're like that big, they're not like, so I'm from Canada, right? And the leeches for me back home are these giant big suckers that live in freshwater lakes, like I've never heard of terrestrial leeches before coming here, right? And on one end of the leech is the foot, and on the other end of the leech is the mouth. And they sort of kind of like inch up you while they look for a nice spot to, suck your blood. And then what happens is that the foot is like a suction, and the foot attaches first. And then what happens is the mouth comes in, it injects you with some kind of, I don't know, venom, toxin, similar to the thing mosquitoes have, where they basically just numb your skin so you don't feel their fangs digging into you, reaching, your nearest capillary or whatever. And this one happened to happen to crawl up someone's face, right? You really can't feel them on your face. It's quite unnerving to look in the mirror and see one on your cheek and go, oh my God. But yeah, so it crawled up her face and into her eyeball. And apparently, it seemed like the person, so the researcher who I pulled it out of her eye was quite used to this. So the thing is when you get a leech in your eyeball, so fun fact, folks, the foot will attach first. And the key is that if you want to have a hope of getting this thing out of your eyeball anytime soon, you need to keep it moving because you need to prevent the mouth with the fangs from getting a hold. So it takes a little bit longer for the mouth to actually dig in. You do have, capillaries in your eyeball. And so the key is to keep the leech moving. And so she felt something in her eye and she thought, well, there's leeches everywhere. There's a good chance this is a leech. So she just keeps rubbing her eye and, can you have a look? Yes. Okay. We looked into her eye. Yep. Sure enough, it's a leech. Okay. We got to pull it out quickly. And that's exactly what I did. She was rubbing it until I was ready. I took a pair of tweezers and I literally just grabbed the thing and pulled. And I swear, like, your eyeball must have like a bit of a mucous membrane over top of it. The membrane came with the leech. And then when the leech snapped off, the membrane like snapped back into her eye. And yeah, it was interesting. And so here's the thing though, is if you're too late, right? So if that leech had gotten its head and its fangs in, you cannot pull that leech out without risking like permanent damage to your eyeball. At that point, you just have to let it suck itself dry. So, it was pretty much a matter of time of get this thing out quickly. Otherwise, we just have to leave it there until it's done. So it's the lesser of two evils at that point.
Wow.
So anyone out there thinking of a career in science, like head to your local university and you too can share wonderful story.
Come and do some chemistry. There's no leeches in the laboratory.
Yeah.
So when we in the first session, the 22/7, when we asked whether you ever had an experience doing a research of getting dirty or it being smelly or something, I would have thought a leech in the eye was probably falling into that category.
Yeah, see, this wasn't, so it was when you guys asked the questions, I took that to be my PhD research. So this was a job that I had done as a backpacker. So I figured it wasn't quite the same. I did do some field work for my own research. I did go out into the grasslands of Queensland and try to sample some rust on wild grasses. So I did actually get to go outside and didn't spend my entire PhD in front of the computer, just, 90% of it. But yeah, no, this was just a totally different bag of tricks. Yeah.
Yeah, and that's a fantastic story.
Actually, I realized I got the city wrong with the Kazakhstan train. Actually, it was Aktau to Aralsk. That was my bad.
That was what
I forgot.
That was
it's
what
been a
confused,
while, so I've forgotten the names.
That was what confused myself, Annie, and Joel. I think
Oh yeah. Sorry, guys, my bad. Yeah.
So let's move over to a couple of minutes of just free for all questions. That's over to you. There's a couple of comments coming through from comments from YouTube and Facebook, and when they do, I'll pop them up. But is there anything else you want to know?
What are you up to now, Michelle? Now that you've submitted your PhD, what comes next in the life of a scientist studying fungus?
Yeah, it's interesting. I'm sort of at a bit of a career crossroads. So at the moment, I have refused any and all job offers because I kind of want to regain my sanity. A PhD has sort of a way of sucking out your soul and your sanity. And, I really kind of needed a bit of a break. So I sort of told myself, okay, when my PhD's done, I'm going to give myself, up to 6 months to just refresh, recover, so that when I go into my next career, I can give it the full attention that it deserves. So that's sort of what I'm doing at the moment, is not much, if anything. I was hoping to spend that time. I love to go free diving. I was hoping to do like camping and traveling, and now all I get to do is basically play video games and do science podcasts. So I am planning to do a motorcycle trip though pretty soon. So that's actually what happened when I was in Kazakhstan. We'd taken a motorcycle. It was 21 years old, the 21-year-old BMW F650, and we rode it from Germany to Mongolia, applied for graduate school in, where was it? Azerbaijan. We applied for graduate school in Azerbaijan because we got stuck there due to visa issues. And we thought, oh, there's internet here, let's apply for grad school. Sold the bike in Mongolia and then started hitchhiking south through China. And when we were in the middle of China, that's when we got our acceptances for Australian grad school. This is my partner and I. And then we thought, oh, cool. Okay, so we just kind of continued south through China and Vietnam and then made our way to Australia. Now I'm here. So now that the PhD's done, the plan is to do another motorcycle trip. COVID is obviously throwing a giant spanner in the works. So at the moment, it's just, I've upgraded to a bigger, more touring-worthy motorcycle. So it's me sort of learning to handle a bigger bike, slowly gathering our camping gear. And so hopefully, when the restriction ease a little bit, we can just head off into the backcountry for a couple of months and just sort of, enjoy ourselves being in the backwoods. Yeah.
Anything else?
I was wondering, like, I've heard, not from, like, from astronomers who work on supercomputers, that there's like a whole thing about like booking the time to work on the supercomputer. How did that go for you? Like, yeah, just having finished a PhD, how far ahead did you have to plan? Like, did you mine Bitcoin on the side? Like,
I wish. Yeah, so you're absolutely right. There is a scheduling system. And so at the University of Sydney, where I did my PhD, we have a program called PBS Pro, which probably a lot of people who do bioinformatics will be familiar with. And this is an automatic job scheduler. So the thing with bioinformatics jobs is you don't tend to have too many very large computationally heavy jobs. You tend to have sort of a lot of small, medium jobs. But these are all jobs that still require too much power for your standard computer, for example. And so essentially what you do is when you're going to run a job, the first thing you do is, you check the documentation for the people who've actually written the program, and they give you an indication of how much time, memory, power, whatever you need to actually process this job, right? And so that kind of gives you a bit of a ballpark. And then usually what you do is you run a couple of test runs on your program to sort of see, okay, how long is it actually taking? How much memory is it using? And then when you have an idea, you have to sort of pre-schedule what your requirements are. And then it's actually really easy because PBS Pro handles most of it. So you basically say, here's my job, here's the resources I need. And then the program just kind of automatically shuffles around people's jobs in space to sort of fit it in. So really small jobs. I'm going to say anything that would maybe compute for less than 48 hours would probably start running within about 10 to 15 minutes. Anything that would run for longer, say, I've had jobs that had to run for weeks at a time. They might maybe take a few hours to a day to get started, but I generally never had to wait longer than a day or two to actually get something running.
it's like when you had to, back in the days where you had to download like a whole season of a television show, not just get the episode by episode each week. It would just like, you'd be like, yeah, I should just leave this on overnight and go do something else.
Yeah, pretty much just made to order. And it's actually really great because like for, sometimes when you're doing a PhD, you're doing something so intense like that, you have a problem when you're not working. You get this like extreme guilt. You're sitting there watching a TV show and you're like, I should be working right now. Oh my goodness. It's really great because when you're running, you're running jobs that take a week and it's like, it's Saturday night, but it's like, well, technically I'm working because my job's running right now. So I'm actually getting stuff done. So awesome. It's really great for like satisfaction that you're actually making progress.
Okay, in the interests of time, I'm going to say thank you to everyone, and just what an absolute pleasure it was to host all of you. Very quickly, you will get an email from me to say, please give me some feedback, and in that form you can say if you want some giveaways. I'm very happy. The first 10 people of each of these Science Week shows will get some cool stuff. But before we wrap it up, are there any pluggable things that you all want to plug to people listening? I'll start with you, Michelle.
Sure. So I guess I run an Instagram where I'll put, fun biology things that I find or interesting tidbits from my life. At the moment with lockdown, the only thing I've been able to do is go freediving. So it's, completely full of fish pictures. But if you're interested, you can find me on Instagram at Michelle's_bio. So Michelle's Biology. So MICHELLES_BIO.
Alice, any plug things?
Okay, it's National Science Week, so I'm going to plug a National Science Week event that we're running at the University of Sydney in partnership with FBI Radio, which is a great community radio station in Sydney at 94.5 FM or online. We're running a project called Live from the Lab, and basically we've partnered 6 researchers from the University of Sydney and from different schools across the science faculty. With 6 local artists from New South Wales. And these artists have composed brand new pieces of music inspired by the science and the research done by each of these people. And we are playing these tracks for the first time ever at 8:15 a.m. every weekday morning of National Science Week. So we started on Friday, we're back on Monday. We're talking solar physics and hearing from Associate Professor Anita Ho-Bailey and Setwan, an awesome musician, on Monday. And the scientists hear the music for the very first time live on air. So you get to hear it at the same time as them and to hear their reactions. So I hope that some of you will be able to tune in on Monday morning.
Annie, plug.
Yeah, well, as always, check out the podcast, Space Junk Podcast and Space Cafe Australia. But this week for Science Week, I'm also partnering up with the Botanic Gardens. They invited me along to do a great space debate on Thursday morning, which is going out to all of the kids across New South Wales or elsewhere, I guess, who are in lockdown and learning from home. You can tune in and watch me and some other far more qualified people talk about, should we be going to space? Should we be solving problems on Earth? What does that look like? How do we do it? Ethics, all of that stuff. So that's on Thursday morning, and you can just look up Botanic Gardens Great Space Debate on your search engine, and you should find it.
Thanks, Annie. And Joel, any plugs?
I think I stuck my plug into my introduction before, but go and subscribe.
Because you're professional.
Go subscribe to the Cosmic Vertigo podcast. It's Corey Tutt and Carly Moon hosting a brand new show for us, and it's super cool. It's a podcast unlike anything we've made at the ABC before, and I'm really excited to hear what everyone thinks when it's out in the world. So thanks, everyone. Yeah.
All right. Thank you so much, team. What an amazing fun time. That was really great. Thank you so much. I appreciate it. And we'll see you all in and around. Thank you so much.
Thanks, Tom.